Okay, this is crafting a scientific story to fit your audience. The first in this year's How-To Talks series. Is anyone familiar with the How-To Talks? Excellent, because we get to explain them then. Okay, so the How-To Talks are a series of talks that are housed here at the library every fall, where postdocs give a talk about how to do something. And usually the talks either have a scientific technique focus or some kind of career improvement, career tips kind of focus. Okay, so they are now until just before Thanksgiving. If you want to see the topics, we have them listed over here. There are some great ones coming up this year, so I encourage you to come. Lots of the postdocs have given How-To Talks before, so they're very great, good speakers. Better than I am at this moment. But I'm Stacey Wahl. I'm the liaison to the Basic Sciences in the School of Medicine here at Tompkins McCall Library. So out of all of the How-To Talks this year, this is the only one that is not given by a postdoc. This is given by me to introduce the series and to get things going. So today we're going to talk about crafting a scientific story to fit your audience. Do you want to know why I know what I'm talking about? To get you to buy into what I'm saying? That would be helpful. So I woke up today and I thought, let's talk about science. Okay, so I am a neuroscientist by training. I did my Ph.D. in New Jersey. I did a postdoc as part of the ERACTA program here at VCU prior to coming to the library. So I have lots of experience both in teaching, giving research seminar talks. I gave a how-to talk. So I have done all of these talks, and we've also used some references to help inform the talk today, which will be listed at the end. Okay? So, to begin, we're going to talk about our learning objectives. Okay, so by the end of today's session, you will be able to deliver a story in an engaging, informative way by identifying your audience, understanding how to develop a central theme to the talk that you want to deliver, and then tailoring your story to multiple audiences. Okay, so we're going to walk through each one of these steps, and how we're going to get there is by answering these questions. So first we're going to spend some time talking about who you talk to. When you're giving a talk, who are the types of people that you talk to? Okay? How do you tell a story? What is the central theme of your story? And then what does your audience need to know to understand that theme? Okay, then we're going to talk about how to keep your audience invested, because hour-long talks, or talks of varying lengths, attention can vary. If you're like me, I struggle to stay awake in anything that's longer than 10 minutes, no matter how interesting the topic is, and that's okay. But we'll talk about how to keep our audience invested, and if, reality, something happens and somebody checks out for a second, how to get them back in quickly so that they don't lose the point of what you're talking about. Okay, then I'll ask you if there's anything else that you want to cover, and then we'll be done. Sound good? Excellent. Okay, so first, these are the 4 types of people that we're going to talk about today. We're going to talk about your lab members. We're going to talk about conference attendees and how that might differ based on the type of conference you're going to, whether you're going to a small conference, a big conference, something that is for your field specifically, or something that's larger. We'll talk about students, both at the undergraduate and the graduate level. And then we'll spend a little bit of time talking about the folks that you might encounter on search committees if you're going to look for a job. Okay? So the first thing that I'd like to do is to talk to you about what you're interested in, because today you're my audience. Okay, so if you could pull out your devices and go to menti.com. I'm going to give you a code in just a minute. That will... Okay. So if you go to menti.com and put in the code up at the top of this, and just answer the question based on who are you most interested in learning about talking to you today. Okay. Cool. There's too many people in here for me to wait for everyone, or to count you to make sure everybody answers, so I'll just guess. right, so if we have anybody else who's answering, it kind of looks like most of what we're interested in is conference attendees and students, okay, which is perfect because we will talk about both of those, but it's helpful for me to know what you're interested in, okay? So let's go back into our presentation, and we're starting talking about our lab members, okay? And so these guys are important because they can be your first test audience, right? Because these are the people who are going to be the closest to your project. They're going to know the most about your work, or more about like the things that you're doing, where you are, because you see them hopefully every day, most days, okay? There's going to be less of a variation in terms of the knowledge base. It's still going to vary because you could have everybody from an undergraduate up to your PI within the lab, but what they know will be more focused, at least to your work, because everybody is hopefully working on something similar, okay? They also might be the folks who are most interested in the details of your experiments, especially they're the ones who are going to ask you, like, what control did you use? Why did you make that conclusion? How did this experiment lead to the next experiment? Because they're really helping you to build your body of work to progress towards being done, okay? But because they're the most familiar to you, it might also be one of your more informal audiences, okay? I've had lab meetings that have been in a break room type area where everybody is eating lunch and we're just talking about something that we have to talk about, and it's not really a formal situation, okay? Conference attendees, on the other hand, have a wider variety of knowledge and skill level. It's generally a more formal audience type. They're expecting something from you. You're usually standing in front of them, not eating a piece of pizza while you're talking, okay? Time limits can vary for the type of talks that you'll deliver at a conference, but those time limits are very strict, right? Especially if you're in a session of lightning talks, no one wants to be there on the 7th talk that's 2 minutes over and you're waiting to get out because you want to eat, right? These folks, depending on the scope of the conference, they will all be less familiar with your work, but how familiar they are is going to vary. Okay, so we're going to spend a lot of time talking about one particular area of research that I'm familiar with, and we'll address that more when we get a little closer. Depending on the conference, they may be interested in your experimental details, but they might be less interested, right? And we can spend some time talking about the different types of conferences and how that really applies. Okay, so students have different skill sets at different levels, graduate versus undergraduate, but even like intro biology versus one of your more advanced things. Okay, strict time limits. Have any of you ever taught before in an undergraduate course? Have you tried to go over? Do people get straight up leave? They're like, sorry, we're done. Okay, so I think conferences have strict time limits, but I think your audience is more polite about getting up and walking out than a group of undergrads. But you have places to be, so it makes sense, okay? That's something that you need to be aware of when you're teaching, is how to stay on task and end on time. In general, students, depending on what you're teaching, they might be more interested in the outcomes of what you're talking about, the facts, what we've learned, than the actual techniques of how you got there. And when I say that, I'm thinking about like an intro biology course where they're trying to understand what photosynthesis is. They might not need to know how you got there in order to understand what it is. But if it's an upper level class in molecular techniques, then how to do a Western blot is incredibly important, right? So it's the kind of thing you have to think about where you are and what you need to then talk about. And then search committees. So search committees are the folks that are going to interview you for jobs. And so they'll differ depending on the hiring goals of the university. So if you're interviewing at a research university, they're going to be interested in how you develop a research program, right? So that's when your experiments are incredibly important. How are you as a scientific thinker going to develop the next 20 years of your career in science? If the university has more of a teaching focus, the job interview talk, the job talk is your opportunity to showcase how well you can teach someone, okay? And then outside of academia, but I don't know anything about that because I work here. But the Postdoc Association has lots of opportunities to learn about how to get jobs in academia or outside of academia, okay? The good thing to think about when you're thinking about search committees is that they're assessing you as much as they're assessing your work. So the way that you speak, the time you spend putting into the talk, if they give you a prompt for the talk that says, we want you to talk about the future of higher education, and you do one slide on that and 45 minutes on your work, that's going to say something to them about you as a speaker and then you as an employee, okay? And again, The background knowledge of these folks is going to vary depending on who is on your search committee. Oftentimes it's people from the department. Some universities may want an HR representative to sit on every committee. That person would probably be outside of your field of work. And so you have to consider that when you're thinking about what you're going to talk about and how you're going to talk about it. Okay? And that brings us to, how do you tell a story? Okay, so we've thought about the 4 different personalities or 4 different people that we're going to think about. And so now I want to spend some time really thinking about, how do you tell a story? And then how does that change based on who you're talking to? So we're going to watch this video, and that's really what I want us to think about while we're watching it, is how do we tell a story? Okay. Because I watched it earlier, that's why. Okay. So, we watch that, we're thinking about telling stories. How do we apply that to what we do? We're obviously not going to be like telling stories about monsters or anything like that, but we do need to think about, what does our audience need to connect to our story? What do they need to know? What do they need to, how do we get them to engage with what we want them to talk about or think about? Okay, so we're going to go back to this guy. So it's the same code that you used before. So I just want you to think about and throw up some words that come to mind when you think about what your audience needs in order to buy in to what you want to tell them, regardless of who it is. You can specify in the parentheses if it's something that's like, students absolutely need this, or lab members need that if you want. Let's just see what you think. So something they can relate to, context, why we value it, what makes it interesting. It's important. If you're trying to tell them a bunch of experiments that you've done, but you haven't convinced them to care about it, it's going to be really hard to pay attention. Okay? Purpose, excellent. Why is it cool? Yes. Connecting the dots. Who said connecting the dots? What do you mean when you say that?
So sometimes when I go to presentations, they provide their research and then they provide a different story, but sometimes it's hard to relate the two together. And it helps a lot when they actually say out the connecting the dots part of it.
So they might say, like, here's this disease that's really cool, and I did these Western blots over here, but not necessarily how their work relates to that disease. Is that what you're saying? Yeah. That's something that we think about a lot. Okay, so this is great. I like that everybody's kind of honing in on this idea of why something is important, because that's what we're going to talk about next, is determining why your thing is important. And I don't know, wait, skip ahead, back to where we were. Excellent. Okay. So, we know that we need to connect our audience to what we're talking about. In order to connect them to what we're talking about, we first have to define what we're talking about. Okay? So we need to think about and really spend some time analyzing what the central theme is of our talk. Sometimes when we approach a talk, we'll be like, I have to tell them all this information. I have to make sure that they know all of these things. But if we spend time thinking about how those things relate to each other, we'll be able to craft a presentation that conveys all of the information without feeling disjointed. Okay? And so when we're thinking about our theme, is there a way that we can represent it visually? Will a visual representation help? Sometimes it does. Sometimes you need words. But let's spend some time thinking about that. And then does our visualization change if our audience changes? We're going to look at a hypothesis together in a second and look at a visualization and kind of work through that about how we would change this or what we would need to do for different audiences. Okay? So the central theme that we're going to be working with is mTOR is essential for oligodendrocyte differentiation past the late progenitor stage. Who knows what this means? Perfect. Okay? Does this help?
Yeah, a little bit.
Maybe. At least now we know we're talking about a thing that goes from this guy over
Right.
here to this guy over here. Okay, so if this was, okay, so let's say this is my hypothesis. I'm a graduate student and this is what I study. This is what I studied. That's why I know a lot about it. If you were in my lab, would you know what this schematic meant? More than likely you would, because every single one of you would use it during the background talk of whatever talk you're giving, right? That's usually how it works. You have these standard slides that everybody in the lab uses, and you say, here's my take on oligodendrocyte differentiation, okay? If you were at a conference, would you understand what this meant?
Maybe.
Maybe. If you're at the Gordon Conference on myelin, you'd probably know what this is, because this is a pretty standard way to represent how these cells develop. If you were at the Society for Neuroscience and you were an electrophysiologist, maybe not. Maybe you'd be like, that's some cool lines, there's some circles, I don't know. Okay? If you're, do you know what I mean by this line right here? What do I mean? Somebody tell me. Yeah. Inhibition. Inhibition, right? If you're a first-year biology student, do you know what that line means? If you're an English student, do you know what that means? Like, what's this upside-down T that we're throwing out here? Okay, so we got to look at that, because this would be something that as a fourth-year graduate student, I would assume that everyone knew, because I was so deep in my project that of course you know what oligodendrocytes are. Of course you've spent intimate time thinking about mTOR and how it's important and where it's important and what it looks like on a Tuesday. You have. But that's really not the case. No one's done that. So that's what we need to think about when we're thinking about our theme, is how does it apply to the people that we're talking to? So this would be a great visualization for our lab. It could work for a conference with some more information, and what would that information then be? Okay? So, we kind of talked through the answer to that question, so I might skip this poll, but if we just want to raise our hands, who would be or somebody tell me, who would understand this the easiest?
Lab members.
Lab members, okay. And who would have the roughest time understanding the hypothesis without more information? Students, or maybe your search committee. You know? If you think about an undergraduate biology department that has 30 faculty members, how many of them do you think study this? I don't know, right? So we have to think about that, okay? So then what does the audience need to understand your theme? Now that we have our theme, they need the purpose that we talked about in our word cloud. They need the background. They need to know why we go from one cell that has 2 branches to a cell that has a bunch of branches and big old globs. And then they need to know some of the methods. Big caveat with the methods is, again, depends on your audience, how much of that you share, how much detail you go into. But these are the 3 main things that you want to think about when you're thinking about your audience. How much of this do they need? What do they need to understand it? What level can you talk about it at? Okay? So, I'm going to drink again, because I talk a lot. Okay. So we're thinking about our hypothesis of mTOR and oligodendrocytes. Why do we care about this? Okay? You don't know. I know. Okay? So here, we have a giant squid axon that can conduct neuronal impulses pretty fast, but it's big. It's 500 microns. So it's half a millimeter. Yes. KJ knows. He told me. That's how big it is. Okay? Then we've got a frog. Same conduction speed. The axon diameter is now 12 microns. Okay, so what do we learn from that? The conduction goes faster. The conduction speed is the same. But the axon itself, the diameter is much smaller. The way that you accomplish that is with myelin. So you need myelin in order to conduct neuronal impulses quickly. Why might that be important? We have a lot of neurons in our body, like 90 billion. What if all of them had to have an axon diameter of half a micron? If you wanted your foot to move quickly, you had to have a big old thick axon in your leg. What could be a problem with that?
Space.
Space, right? This allows for more complicated organisms to do stuff more quickly. Okay? Now you're connected. I think I've sold you just a little bit on the importance of myelin. Okay. We also have this. This is a picture of a giant fish trying to eat a smaller fish. Okay? Does this make you want to know how this relates to myelin? If I just showed you this picture and said, myelin's important because of this guy. Yes? You want to know that myelin evolved either for the big fish to eat the little fish, for the little fish to get away. So there's this theory of predation, escaping from predation, or being better predators, which is why we developed myelin. Beside the point, but if you are giving a talk to undergraduates who need to know biology in order to pass, which one would you start with?
Yeah, the right one.
Yeah.
The one on the right.
Giant fish, conference, what will we start with?
Left.
Oh, we think the conference wants to know more information, all right.
No.
What about search committees? Right, giant fish. I think the giant fish wins every time because it's cool, right? But if you need to connect folks to why myelin is important and what specifically it can do, the image on my left, your right, or that one, left, is much more informative than the one on the right.
It's a good follow-up
Right?
to the first one.
It would be good to have them together. So say like crazy picture of a fish and then get more into it. But it's important to think about what's going to grab your audience's attention first. Okay? Then we think about, now we're going to think about the background that we need to know to understand this. I gave you a little bit on what is myelin because I can't help myself. But let's go back to Menti and see if we can do another code. The code's going to be different this time. So go ahead and try to find the spot where you can enter another code. And we'll just vote on in order to understand this hypothesis and be engaged in our talk, what does our audience need to know? Or how important are the answers to these questions? Okay, so I think that we can agree that the top 4 questions are all going to be important for most audiences. Without knowing what mTOR is, it's going to be hard to get someone to care about the rest of the talk, right? If they don't know what myelin is, then oligodendrocytes aren't important. And if you don't know what oligodendrocytes are, well, then you're just like, what am I doing in this talk about these squiggly things? What's interesting is the value of what does mTOR do in other cells. I think that there is one audience that would want to know that information. Can you think of an audience who would want to know that?
Research community.
Search committee?
Students.
Maybe.
Lab members, for sure.
And so why would your lab members want to know that?
In case it impacts their work with other cells.
Right, let's say it's a whole lab about mTOR. But also, they're the folks who are going to help you brainstorm what to do next. So they're going to be the folks who are most interested in what you're doing. So if they can say, like, hey, mTOR is really important for autophagy in the gut, does it have a similar role in the brain? Because we all work in the brain and know that we make so many more cells than we need, right? They're going to be the people who are interested in doing that kind of brainstorming with you, and that's where you might want to include that information. What mTOR does in other cells might be an also great way to get conference attendees more interested in what you're talking about. If you're at, say, SFN, and you're talking about myelin, but you know it's really important for this subset of neurons, you could throw that in there. And then anyone in the audience who studies those neurons is now your new best friend because you talked about them. Okay? Search committees, I think, I would guess they wouldn't care. But it depends. Most of the time, I think that's a more basic level talk, a more like, let me show you what I can talk about, because it's more about you as much as it's about your work. Okay. So that's how that's a way to assess what we need to know, what we need to put into the background. Okay, and then I turned this off so I wouldn't move away from our pool too early. Okay, methods. If we're giving a talk where we're going to include the methods, it's helpful to think of a way to visualize them so that your audience can understand them quickly. So oftentimes methods are very detailed. The nuance of your method is very important. And it's also something that is kind of hard to hold all in your brain at the same time because it's complicated. So when we think about the methods that we're going to talk about, how can we make it easier for someone who hasn't done those experiments to understand why we made the choices that we made? And so this is an example of how to do that. This is a timeline of time points for experiments. So these are the 3 time points that we looked at things. This is why we looked at them. Okay, so the onset of myelination, right after the peak of myelination, and then an adult. Okay? And then just to remind folks of what's happening in the organism at the time, we have this fun diagram here, and then a timeline of the age of the animals so that you can orient yourselves quickly, right? So then I would explain this at the beginning of giving data, and then I'm talking about some PCR that I ran or a behavioral test that I did, but it's got this up in the left-hand corner. What do you know if that's up in the left-hand corner?
And that's where you're at in the timeline.
Right. Whatever data I'm talking to you about happened or was collected at this time point, because I've already explained to you that these are the time points that we're looking at. So if you look down at your phone because you got a text message and you look back up and you're like, what in the world is she talking about now? At least you know that we're talking about something that happened at P14. Okay, and then you can change it to reflect what you're working on. This is a nice way to bring people in, keep them along, keep them going through what you're talking about. Another way to show your methods is to just have a cool diagram. This was adapted from another graduate student in the lab. This is a base method that we all use to collect cells. So if you're giving a talk to lab members or a really specific conference or a search committee at a research university, how you do your methods is important. Being able to walk them through it with pictures is helpful because it's complicated, and so if I was to explain this to you with only words, it would be kind of boring. Interesting to me, hard to tie into, hard to get hooked by if you haven't done it before. Okay, so this could be something that you only make for the talk that you're giving. Okay, so also be prepared to think about needing to make new stuff based on your audience. Okay, so we're not always going to use the same way to explain our methods to different audiences. And what we want to do, the question that we want to answer is, what does our audience need to know about my methods? Not necessarily, what do I want to tell them? What am I really excited about? Like, I'm really excited about the differences in time. If you put them on the shake, what happens? But you don't need to know that, right? Okay, so now that we've sold them on why we're doing something, given them the information they need to understand it, and told them about how we're going to do it, what do we do to keep them invested? Okay, so this is usually the beginning of your talk, and then you're like, okay, now I'm going to tell you a bunch of stuff. How do you keep folks invested and not checked out? By providing a roadmap, okay? So there's one caveat to when I would strongly advocate not providing a roadmap, and we'll talk about that at the end. Okay, so this is for most things, and then we'll talk about the exception to that. Okay, a roadmap can orient your audience to the outline of your talk. This is good for folks who like to know what's coming, who like came into a session and want to make sure that they're in the right spot and they should really sit there for the next 50 minutes. Okay, this gives them a preview of the content that you're going to talk about, which is a way to engage them before you even tell them the point. So before you even introduce your hypothesis, you can say, I'm going to talk about these things. And then they're like, I want to stay to figure out what happens when she gets there. And it also provides you a framework to keep referring back to give people the opportunity to come back in, to be realistic about attention spans and audience behaviors, and provide a way for folks to continue to see where you are in your talk and how they can get back into the content if they've taken a break. Okay? Roadmaps will only work, though, if you use them the entire time. I've seen lots of talks and given a few where you introduce this really long outline at the beginning, and then you put it up at the end. And in between, a whole bunch of stuff happens. And you're like, as an audience member, I find that confusing, because I'm like, how do I know where I am in the talk? Is this person, like, on track, or are we going to be here for the next 6 years? I don't know, okay? I did say there was one exception to providing an outline. Does anyone have a guess what I might mean? Perfect, okay? 10-minute talks, okay? 10-minute talks are something that you'll give at a conference, you'll give at symposia. They do not need an outline, okay? 10 minutes is a reasonable length of time to expect people to pay attention to one topic. We can talk about how much information you can cover in 10 minutes because I think that that's another big thing. But if you spend 2 minutes explaining your roadmap and 30 seconds referring to it on every third slide, you've then lost 4 minutes of your talk to explaining your talk. And then what do you're down to that much lower content that you can give them. Okay? Okay, so let's talk about examples of roadmaps. Okay? You know that mTOR and oligodendrocytes are my jam, so that's what we were talking about before. This is another research roadmap that you can use, okay? What do you think I would talk about in this thing?
Question marks.
The question marks, right? Can you tell the order that I'm going to talk about these things in? Right, there's numbers, okay? So you don't need to know any of this stuff. it doesn't, I mean, it's great that I drew the structure of the receptor for you and that there's 2 ligands by it, but that's not really important for you to understand what I'm going to talk about. You know that I'm going to talk about how mTOR relates to whatever's happening here. Then we're going to come around over here and look at this stuff. And then we're going to look at this stuff. I can refer back to this throughout the whole talk by animating different parts in and out. And you can stay oriented to the overall picture because I'll keep coming back to it and be like, now we tested whether mTOR was important for the interaction between YY1 and HDAC. And we found it's not. So it's a way to introduce hypotheses and conclusions all wrapped up in one thing to keep you focused. Okay? Teaching. This is something that is great for teaching. Okay? So here at the library, we teach a lot of classes on how to do literature searches, how to structure a search, how to take a research question and turn it into something that you can put into a database. And so John Cyrus, our liaison to the clinical side of the School of Medicine, developed this roadmap. And what's great about it is because it's very easy to orient the students as to where we are in the process, right? And it's just by changing the color of this. So if I made this one yellow, we would be talking about how to refine the search, okay? And so this is really great because it tells you at the same time where you're going and where we are, okay? It's easy to pay attention to and to get into, okay? This one takes a little bit more explanation, but it's also great. This was from a job talk for a recent hire within the library, and he was tasked with talking about teaching multimedia, okay? And so he comes in and he's going to explain different teaching scenarios, and he's got different students who are in this teaching scenario. And so he spends a little bit of time talking about the students, providing you some background, and basically getting you to the point where you understand how much time they have and how much money they have to devote to their studies. Okay? This roadmap I like because it combines some background and takes a little bit to explain, but then when we get to a new class or a new assignment, I could show you the colors of the students and change these around, and you would know clocks meant time, piggy banks mean money. So we could talk about the money and the time of these students without having to walk through each time what it means, who they are, and then we don't have to put like dollar signs up there or specific amounts of money or specific amounts of time. You don't have to do math. Although when I was talking about this slide with somebody, they were like, why is he not 3? Why does no one have 3 clocks? Why do we go from 2 to 4? And I was like, I don't know, man. But this is good because it gives you a visual representation of who we're talking about and what's important about those people. Okay? So, we think about our roadmaps, we think about how we're going to get people to invest. We do next. Oh, yeah. Then we're at our last point. What do we do next? What else do you want to know? We have a good bit of time left, so this is the bulk of the content that I wanted to deliver to you. Okay, we talked about who our audiences are. We talked about the different things that we want to give them and how that might change on who they are. But what didn't we cover? What did you come here wanting to know? Yeah?
Could you speak a little bit more on the invest bullet point about other creative ways to make your story a little less dry than what a general audience might think, other than just the outline?
Okay, so another way to make your story less dry. I think that brainstorming the connection to the why and putting your time into the why is important, because a lot of biomedical stuff that you're doing in a lab if you're funded through the NIH, you have like a disease model that is the overarching thing. So you could talk about why that disease is important. Who has that disease? What are the demographics of it? What are the consequences of it? So connecting it to that human aspect. What else do we do? Another thing that you can really do that makes a huge difference in how interesting an audience finds your data is to be excited about it. So a lot of times when folks talk about their data, they're like, So I did this Western blot, and you can see that mTOR is down on day 3, but it comes back up on day 4, and then we looked at that, and it's quantified here. Do you think I cared about that experiment? No, it doesn't sound like I did. But if I come in and I'm trying to tell you that this is the greatest thing that's ever happened, it is the coolest thing. mTOR is by far the best kinase in the history of the world. Whether or not you agree with me, you're at least like, what is going on over here? I need to pay attention to it. So the enthusiasm that you can bring to your science is going to sell it just as much as how cool the science is. Because everything that you do is cool. If you're excited about the result you get when you get it, that's the level of excitement you should convey to the audience. No? Yeah.
What is kind of your method for finding like a really good metaphor for something when you're talking to like introductory biology students, for example, somebody from a different discipline who might not have that knowledge? What's, I guess, the best way to craft a metaphor there?
That is a really good question because I am terrible at metaphors, but it's okay. My husband is an engineer, and whenever he explains something to me about the power plant, he compares it to cars. And I think it's because he understands how cars work. So when I think about developing a metaphor, I try to think about something else in my life that I either know a lot about or I'm really excited about. And that's where you're going to have the most information to be able to build the relationship between two concepts that makes sense. I think, what do I talk about a lot? I talk about marching band a bit, just to like showcase that I'm a nerd in all facets. I don't know. Yeah. I really just try to, like they said in the video, go with what you know. Anybody else? Yeah?
If you are giving a 10-minute talk at a conference that is extremely broad, so people that aren't in science to experts in the field, should you focus on making sure that the people that aren't scientists understand your data, or should you sacrifice some of explaining some of the background to those people to make sure that the experts understand what your data conveys and why that's important to the field? So like striking that balance.
I think, what do you want your audience to know about your data? I would answer that question first. Is it important that they understand the intricacy of the experiment or the nuance of the result? Because if you're getting something where the result is kind of nuanced and you need to really understand the biology in order to understand that nuance, then you might want to spend some time there. But if that nuance isn't critical to the overall conclusion of the experiment, then for a 10-minute talk, you might just not talk about the nuance. I think it's really important that when you're considering your audience, not everyone in your audience has to understand every single word that you say, but they should all walk away with a good sense of what you were trying to tell them, so that they can then follow up with you if they have more questions about that nuance.
Can you speak a little bit about your perception of incorporating this interactive technology with the different audiences?
Yes. So, did you guys like doing polls?
Yeah.
Did you guys do polls when you were in school, in undergrad? Okay. Some yeses, some nos. Okay. So, I think it's good to try and incorporate audience feedback when you can, if you're comfortable with doing something with that feedback. Okay, asking a question and then continuing on is not really using the technology to its fullest, I think. I think if you're going to ask your audience something with some of these interactive measures, you should be willing to then use that in some way. Okay? Use it as a way to build a relationship with your audience. Use it as a way to focus on different parts of your content. Whatever you want to do, but asking a question and then not incorporating the answer could make your audience disengaged. I think working in groups is awesome, and when you can work that into a talk, it's great. Not always practical for research talks, but teaching is a fantastic way to get folks to engage, because you're asking them to then contribute to the content of the talk. And that empowers audience members to feel like they're part of the team and makes them want to do something, right? And you learn better if you do stuff. Does that answer your question? Anybody else?
Can you tell us more about this how-to talks? Like, how does it work? I mean, not how do you have to prepare the talk, but actually the entire series. I believe you have there's a timeline that you have to propose yourself at some point. And is there like a competition that you select from the pool of people?
So so the how-to talks, this is the 4th year of the how-to talks. I think so. So this is the 4th year of the how-to talks, and the timeline of applying, accepted, and stuff has actually changed a little bit this year. We asked for applications earlier, so we accept applications for the how-to talks in the beginning of at the end of the spring semester, beginning of the summer. And then we spend the summer going through it, building all of our promotional materials, and then if you're selected for a talk, we work out dates. They're always Mondays in the fall, and you give a practice talk the week before where we work with you to make sure that your talk is a how-to style talk and not necessarily a seminar talk, because they're so different that it really takes time to get into that headspace. And this year we have a competition for the best how-to talk. So the talks are judged by a 3-person panel, and the best how-to talk will be awarded at an award ceremony in December, and they will receive a cash prize. And you have to be a postdoc in order to apply.
What do you take into consideration?
We look at we don't ask for too many things. We ask for a short summary of what you intend to talk about. And we basically look at that just to see if it would fit into this series. If you have a how-to that's relevant. If it's a research talk that's loosely repackaged as a how-to, that's something that we would question more, because we really want these talks to have the audience walking away knowing how to do something. And so we really want to make sure that it's a technique or a career development thing that can benefit the VCU community.
One more question.
Yes.
Do you have a numbers, like you get 100 applications, select 7, or?
So it's not a strict competition. We get 10 or so applications, between 5 and 10 every year. And we try to accommodate as many people as we can, because it's such a great opportunity to practice a different communication style. So it's not really a tough get.
Maybe we try to, if people have done it before, or if they submit more than one, then we try to give as many people as possible an opportunity versus people doing multiple invitations.
For written storytelling, rather than say giving a presentation, how do you convey excitement?
This is a great question. So my postdoc mentor was fantastic at grant applications, but she had a very informal style, and that's how she can convey her excitement. So that was just well-funded. So
Do you just use language like, I don't know, exciting language?
I think I would be compelling when I'm connecting it to the why. So I would use more exciting terminology when I'm connecting it to the disease background or to the relevance within the field. That some of that demographic data, if you're using a disease model, could be really compelling. Specifically, if you're focusing on an audience, like if you're applying to NIDCR, or if you're applying to the children's one, NICHD, and you focus your background on children, that could be really compelling. Another thing that I've seen folks do in order to make their grants stand out is to use images on their specific aims page. So to do something like that to stand out, because most of the time it's just a huge wall of text, and then people in the study section get 30 of those that they have to read, and if yours is the one that has pictures in it, some of these roadmaps that we were talking about before, applying that into a grant. Anything else? You guys ready to go tell awesome stories about all the things? Excellent. Well, thank you guys for coming today. Be sure to look at the sign on the way out to check out the other how-to talks that we have offered this year. And the feedback that you guys have in your hands is for me to do better. So be honest.